This is the first lesson of Chapter 14. We've come full circle now in the class. We're going to be looking at how the code of life, DNA, is used to make proteins. That would not be possible without the structure shown here on the slide. Now there's a lot going on here, lots of different colors and some helices, what looks like DNA, single, double strands. This is what you might think of when you look at a diagram of a cell, that little speck, that tiny little dot. But it's a very important organelle, and as you can see, it's not just a dot, it's got a lot of complex structure. So, do you recall from an earlier chapter in the class, what is the name of the organelle that makes proteins? Well, it's the ribosome. And all cells have ribosomes because all cells, prokaryotic, eukaryotic, plant, animal, fungi, protist, bacteria, every single living thing needs to be able to make proteins. This is the organelle where that happens, the ribosome. So what is a ribosome? Ribosomes are kind of complex. It's not just a little dot, as we usually see it depicted in a diagram of a cell. In larger terms, there are two major subunits of the ribosome. And you can sort of see like it has like a cap, kind of like a mushroom cap, and then this other part, the base. So these are the two ribosomal subunits. The larger subunit is the top one, and the smaller subunit is the bottom one. The larger subunit is made of 33 different proteins and also RNA, ribonucleic acid. So the reason for the different colors is to depict the different polypeptides that comprise the proteins. And you can see that there's both what kind of looks like DNA in there, but that's actually RNA. So it's pretty complex. The smaller subunit, the bottom part that's more in this teal and blue color, is made of 20 proteins and also ribosomal RNA. So it has quaternary structure of a protein because there are more than, it's got the sequence of amino acids, there's alpha helices, beta pleated sheets, so it's got secondary components of the structure, tertiary meaning how it interacts with its within one polypeptide on itself through things like ionic bonds, and then the quaternary refers to the fact that there's more than one polypeptide here forming this structure. In fact, it's what, 33 plus 20, 53 proteins. Crazy, I know, for one little, what you sometimes see is just a speck on the cell. Now, ribosomes are not all created the same. Ribosomes from eukaryotes are larger and more complex than those from prokaryotes. But we won't be digging into those differences. Just appreciate that, in general, usually eukaryotes are larger and more complex, and so are their structures. But we're going to now just focus on the most simplified version of this process of how genes are used to make proteins and how the ribosome is involved in that. So this is actually going to be a two-step process from gene DNA to protein. The first step in the process, large step, because we're going to be digging into the details, of course, the first overall step is called transcription. And this involves the synthesis of a type of RNA called messenger RNA, or mRNA, from the DNA code. Then, once that messenger RNA is produced, it heads to the ribosome, where translation takes place. The messenger RNA is going to be used as a template for transfer RNA, tRNA, to bring amino acids and build the polypeptide, which ultimately becomes a protein. Okay, now this is a very, very broad overview of how genes are used to make proteins. So we are going to be looking at the more specific details for both transcription and translation. Before we do that, as always, let's see how this would compare between a prokaryote and a eukaryote. So on the right-hand side, we have two representative cells. On the top is a prokaryotic cell, and on the bottom is a eukaryotic cell. So first of all, notice the prokaryotic cell is much smaller, simpler, and for sure, one thing you should notice is it doesn't have a nucleus. So where's the DNA in a prokaryotic cell? Well, it's just floating around in the cytoplasm. You could say it's in the nucleoid region. It's just basically the region wherever the DNA is in the prokaryotic cell, but it's just in the cytoplasm. Versus in the eukaryotic cell, the DNA is inside the nucleus, and that's where it has to stay. DNA cannot leave the nucleus in total as like a whole molecule. So for the parts of the DNA that are going to be used to make proteins, that's only going to be leaving in the form of messenger RNA. But before it can do that, there's some processing that has to take place. So where it says in the eukaryotic cell, DNA to pre-messenger RNA to mRNA. So the pre-mRNA is a molecule that has to be further processed before it can leave the nucleus of the eukaryotic cell and then go to the ribosome. You don't see that extra step in the prokaryote. It just goes from DNA to messenger RNA. There's no processing that needs to happen because it's already outside in the cytoplasm. There's no nucleus that it has to go through. But although there are these differences, the mechanisms overall are the same. DNA is transcribed to messenger RNA, which is then translated into proteins. So let's stop there for this lesson so that we can focus in our next lesson on transcription specifically, and then we'll have another lesson on translation.